excited to chair the remainder of this session, which will continue on plasticity. I think very nice continuation of the theme. And I'll introduce our next speaker, who is Kishore Hari. He's talking about phenotypic plasticity.
Thanks, Adam. I would like to start by thanking the organizers for putting together this wonderful meeting. I can honestly say that this is the coolest set of people in a conference that I've ever met. So I really appreciate being here. Uh, as for the talk, I'm also happy that I go after Adam because most of the introduction that I'm supposed to give, Adam already gave. So I'll just quickly jump into the talk. So I'm interested in understanding what kind of plasticity cells undergo during metastasis, particularly through the lens of EMP. And so EMP is basically this spectrum of cells ranging from epithelial to mesenchymal. And while undergoing metastasis, cells at the primary tumor have primarily epithelial phenotype. During migration, they have more mesenchymal or what is known as a hybrid phenotype, which has the properties of both epithelial and mesenchymal, what Adam referred to as intermediate EMT phenotypes. And there are some really cool properties of these hybrid phenotypes listed here in this very simplistic version of a very detailed, complicated set of experiments. So what you see here is a pseudo landscape. Of course, physicists will hate this. I'm sorry about. But the depth of each valley you see here is representative of the abundance. If you take a population, heterogeneous population, then you will see more cells being epithelial and mesenchymal than hybrid. And then you have those arrows on top, which tell you about how easily they change their phenotype, change their expression patterns to convert to different other phenotypes. There are, of course, other interesting properties that hybrid states have, including having really high plasticity, stemness, invasion, and so on. And high plasticity is often taken synonym to them being so-called metastable. And the second part of my talk will basically challenge that. So again, as Adam mentioned, these phenotypic transitions are controlled by gene regulator networks. There is a central sort of motif that you can see in EMT networks. This is what we call a double negative feedback loop or a positive feedback loop. And depending upon which node has a high expression, you either have a mesenchymal or epithelial phenotype, broadly speaking, right? But when you actually want to capture the heterogeneity of hybrid states, you also want to look at larger networks so that you can have more chances of expression heterogeneity. And that's what we try to understand. And the first question that I want to address is what are the special features of these large networks underlying EMP? And when it comes to plasticity, the main characteristic is multistability, and multistability is famously driven by positive feedback loops. But there are a lot of positive feedback loops in these large networks. And what we find is that if you take the network in the form of adjacency matrix, which is essentially telling you what are the direct interactions between the nodes, and also consider interactions which are indirect, mediated through other nodes, and make some cool transformation, you get what is known as an influence matrix, right? So what the influence matrix does is it captures all the influences within each node and gives you this nicely separated structure of two teams, right? So each team can be thought of as a huge coarse-grained node, and the interactions are such that each team is inhibiting each other and activating itself. So we again have a positive feedback loop, but at a higher dimension, right? So this is a very clean coarse-grained structure that we observe across multiple contexts, not just EMP. So over the last couple of years, we have been analyzing networks both in cancer heterogeneity as well as developmental scenarios. And across all of these contexts, we find the team structure in the underlying gene regulator networks. And using various different simulation formalisms, we have been able to verify that when you have such strong teams, then the emergent landscape, the emergent phenotypes broadly take a bimodal distribution. Essentially, you have two very stable phenotypes in the case of EMP, epithelial, and mesenchymal, and then you can have some less stable phenotypes, right? And the influence matrix that I mentioned before is also very useful to identify these interesting patterns. So one of the yeast cell cycle network shows a overall negative feedback loop, which essentially gives you the oscillations required to control the cell cycle. We also found some cool cool signatures in the transcriptomic data, which can only be explained by having teams. So we think that teams can sort of help us get better at network inference. And my friend Vaibhav is also thinking about using teams outside gene regulator networks. So please do visit his poster. He can tell you more about it. So given that these networks have teams, we wanted to understand what their sort of emergent phenotypes look like, what is their plasticity looks like. And we took a very simple model. It's a Boolean model where we are only concerned with what is the sign of each edge, and the expression level of node can either be +1 or -1, right? And we simulate these networks at a large number of random initial conditions, giving rise to a heterogeneous population of digital cells, which looks essentially something like this. Each row is one instance of the simulation, and what I'm showing here are the steady states. And you can clearly see that there are these three classes of steady states here. Majority of them are epithelial and mesenchymal, and a small minority of them are hybrid, right? These hybrid states essentially have expression from both the epithelial team as well as the mesenchymal team. And based on our team structure, this kind of a configuration is not really supported, and therefore their expression is much lower, right? Very clean cut, very simple and easy to understand, makes our life very easy. Now, then we wanted to look at plasticity, right? How cells change their phenotype. And one common experiment that is done to understand plasticity is people take a heterogeneous population of cells, isolate different phenotypes, and then let those phenotypes again repopulate and see what kind of heterogeneity is recaptured. So if you start from epithelial or mesenchymal, most of the cells remain epithelial or mesenchymal. But if you start from EMT, in this case is the hybrid phenotype, you will have a more heterogeneous population, right? So we did a similar experiment where we start from a homogeneous population of cells. Each cell has the same expression pattern. And then to get things started, we just gave them a small perturbation, let them evolve, and measure plasticity, which is how many of these cells did not come back to their original expression pattern. And as expected, supported by the network structure, supported by the theory, epithelial and mesenchymal cells have a very low plasticity, whereas hybrid states have a very high plasticity, right? But there is one problem with this picture, that hybrid states cannot stay stable even for the lowest levels of perturbation. Okay, so they're a bit too plastic, which may or may not be the right kind of plasticity that hybrid cells should have. So if you look at some of these uh, tumor growth experiments or metastatic experiments, where people again start with clones of epithelial, hybrid, or mesenchymal cells and see how heterogeneity evolves. In vivo, what people see is that there is a significantly higher fraction of hybrid states than what our model would predict or what the in vitro experiments would just suggest, right? So we needed to explain this, and there is one way of explaining by saying that in vivo cells are gaining mutations and so on and so forth, but there could be an easier explanation, and that comes from this other observation of hybrid states, that consistently from the initial observations of hybrid states, when people started to look at the expression of different genes, they see that hybrid phenotype always has partial expression of genes across the spectrum, right? And even in CCLE, if you look at all the carcinomas, identify the hybrid cell lines, and look at what fraction of epithelial and mesenchymal genes have partial expression, then you see that most of hybrid cell lines have at least 50% of mesenchymal and epithelial genes showing partial expression. So to incorporate that, we updated our Boolean formalism to a multi-level model, where this was the original two-level model where you have only -1 or +1 expression, and to this model where you also now allow two more intermediate levels of expression. And unsurprisingly, this allows for some states to see partial expression, right? So previously it was completely red or blue. Here you have light red and light, right? One thing to notice here is that the epithelial and mesenchymal states still retain like a very high expression level, and the partial expression is focused only on hybrid states, which is something that we wanted to see. So far, it's a good news. Yeah, so that's what I'm showing here, comparing the Boolean versus hybrid multi-level hybrid states. You see that there is a lot of partial expression in multi-level hybrid states. Another interesting thing is that in the partially expressed hybrid states, most of the cells from the same team in state 1 and state 5 have similar expression, right? So that's again something that is supported by the network topology. Previously, no such state existed. So that's another good news, that we might be capturing some true hybrid state which might be more stable. Another thing that we can capture with this model is that you now see a lot more hybrid states than you saw before, right? Which gives rise to the possibility of more heterogeneity in the population of hybrid states. Another thing that we really like about hybrid states. And then we looked at the plasticity of it, of these new hybrid states, which are supposed to be more stable. Again, perform the same experiment where we give the cells, homogeneous population of cells, a transient signal, and ask what happens to the final phenotype. Do they come back? Do they not come back? And here on the y-axis, I'm plotting the fraction of retention, which is 1 minus plasticity that I showed you earlier. And as you can see, the hybrid states that you start get in the two-level or Boolean model, they are much more stable in some sense than the hybrid states that you get in multi-level. So in a way, these new hybrid states in multi-level formalism are more plastic. Okay, but what things get interesting from here onwards. So now we tracked not only what is the expression level of the perturbed state, but also what is the actual phenotype, because you can have multiple state configurations having the same phenotype, right? And the interesting thing here is that the Boolean hybrid states, they quickly transition epithelial or mesenchymal phenotype. But the multi-level hybrid states, there is a significant fraction, even at the maximum amount of perturbation possible, there is at least like 25% of the initial population that retains its hybrid phenotype, right? And this is something that we see in these in vivo experiments as well, where you start from a hybrid clone, and there is a significant fraction of the emergent population that retains its hybrid phenotype, right? So that's again good news, but it contradicts with what we saw before, that the multi-level hybrid states were supposed to be less stable. So the answer to that is here. So in this particular plot, I'm tracking the distance in the expression level of the initial state and the final state after perturbation. And as you can see, the Boolean states don't really wander off that much, right? They stay in their locality. The Hamming distance is very low, but the multi-level states, they wander off a lot, right? So after perturbing, each state is changing its expression and moving to a different state, but it's still largely remaining hybrid, right? So this is a really interesting phenomena that we name hybrid cloud. And we also tested this with more stochastic perturbations, and we found similar results. But basically, what we are saying is that you have these hybrid states, which is like a heterogeneous cloud, and plasticity happens between these individual state is actually very stable, but as a cloud, as a collective, they are more stable, which could be more fit for metastasis. So the takeaway messages are mutually inhibiting teams serve as design principles for self-fate decision networks, and the hybrid phenotypes can have different kinds of plasticity. We capture what is known as a cloud type of plasticity, what we are calling as cloud plasticity. And the question that we're interested in asking now is, what kind of plasticity is more suitable? So I would like to acknowledge Mohit and Harbi and all the other people involved in this work, and leave you with this picture. So based on my understanding, there are three kinds of plasticity that you can have, and it's really interesting to think about what kind of plasticity is more suitable for what condition. Thank you.
Great. Thank you very much, Kishore. We've maybe got time for one quick question. Joel. I'm prepared to jog. I've been sitting all day.
Hey, that talk was wonderful. Thank you, and enjoyed the plasticity and the approach that you're taking to it. Can I
Thank you.
take kind of a slightly different angle and ask you, approach this phenotypic plasticity as an evolved adaptation and ask the question, does this function successfully either for the cell within the network or for the cells that are cell-cell interaction?
I mean, I think yes, that you could think of it as an evolved phenotype, but I'm not entirely sure how to think about the cell-cell interaction part that you mentioned. Maybe I can talk about it later with you. Thanks.
Okay.